In this video, you will first learn how alleles determine a person's traits and how to predict the probability of inheritance based on parental genotype. Then we will move on to apply this foundation to the inheritance of autosomal dominant, autosomal recessive, and sex-linked genetic disorders. To understand how traits are inherited, we first need to review the structure and organization of human chromosomes. Humans have 23 pairs of chromosomes. Each somatic cell contains two sets of chromosomes, one inherited from the mother and one inherited from the father. Because somatic cells contain two sets of chromosomes, they are described as diploid. The diploid chromosome number in humans is 46. The two chromosomes within each pair are known as homologous chromosomes. Homologous chromosomes are matching pairs that carry equivalent genes at the same positions on each chromosome. One pair of chromosomes are the sex chromosomes, which determine the sex of the individual. Females typically have two X chromosomes, while males typically have one X chromosome and one Y chromosome. The other 22 chromosome pairs are called autosomes. Autosomal chromosomes are the chromosome pairs that are not sex chromosomes. Now, I will define several key genetics terms that help explain how traits are inherited. A classic example I will use to explain inheritance patterns is the ABO blood type system. This trait is important for safe blood transfusions. To avoid a dangerous transfusion reaction, we need to cross-match the donor's blood type with the blood type of the recipient. You will learn more details about blood type when you study the cardiovascular system. A genotype is the specific combination of alleles that an individual has for a gene or set of genes. An allele is a variant form of a gene located at the same position or locus on a chromosome. For genes on autosomal chromosomes, humans inherit one allele from each parent. The gene that determines ABO blood type is located on chromosome number 9 in a specific locus named band number Q34.2. There are three different alleles for this blood type gene: A, B, and O. Different combinations of alleles can interact in several ways to produce different traits. An individual that has two identical alleles for a gene is described as homozygous. Here we see the homozygous genotype with two O alleles. The phenotype refers to the observable traits that result from gene expression. In this case, blood type O is the phenotype that results from expression of the O alleles. An individual that has two different alleles for a gene is described as heterozygous. Here we see the heterozygous genotype with one A allele and one O allele. The interaction between alleles determines which phenotype is expressed. A dominant allele is expressed even when only one copy is present. Dominant alleles can mask the expression of recessive alleles. Sometimes this masking is incomplete, but in many cases, the dominant allele determines the phenotype. A recessive allele is only expressed when an individual is homozygous for that allele. Even though their genotype is heterozygous, this individual's phenotype is blood type A because the A allele is dominant and the O allele is recessive. Another type of allele interaction is codominance. In codominance, two different alleles are both equally expressed. The ABO blood type system produces four different phenotypes based on combinations of three alleles. These include two codominant alleles, A and B, and one recessive allele, O. Type O blood only occurs when an individual is homozygous with two recessive O alleles. Type A blood occurs when an individual inherits two A alleles, or one A allele and one O allele. Type B blood occurs when an individual inherits two B alleles, or one B allele and one O allele. Type AB blood occurs when an individual inherits one A allele and one B allele. In this situation, the alleles are codominant, so both A and B are expressed. Scientists often use a Punnett square to predict how alleles may be inherited. A Punnett square is a diagram used to predict the probability of possible genotypes that offspring could inherit based on the genotypes of each parent. For a simple example, the Punnett square is arranged as a 2x2 grid with 4 boxes. The alleles from one parent are placed across the top of the square, and the alleles from the other parent are placed along the side of the square. Each box in the grid represents one possible combination of alleles that a child could inherit. For example, consider a heterozygous type A parent with genotype AO and a heterozygous type B parent with genotype BO. The type A parent can contribute either an A or an O allele. The type B parent can contribute either a B or an O allele. When these alleles are combined in the Punnett square, 4 possible genotypes appear. These combinations are AB, BO, AO, and OO. These genotypes correspond to the blood types AB, B, A, and O. Each of these outcomes has a 25% probability. Punnett squares are often used to estimate the probability that a child may inherit a particular trait or genetic disorder. For single gene disorders, genetic testing can allow clinicians to calculate the probability that a child may inherit a specific disease. One inheritance pattern is autosomal recessive inheritance. An autosomal recessive disorder occurs when an individual inherits 2 abnormal alleles for a gene located on an autosome. Because the abnormal allele is recessive, individuals with only one copy usually do not have the disease. These individuals are known as carriers. An example of an autosomal recessive disorder is cystic fibrosis. Cystic fibrosis is a disease characterized by the chronic accumulation of thick mucus in the organs of the respiratory and digestive systems. If 2 heterozygous carriers have a child, there is a 25% chance that the child will inherit cystic fibrosis. Another inheritance pattern is autosomal dominant inheritance. An autosomal dominant disorder occurs when one abnormal allele is enough to produce the disease. Because the abnormal allele is dominant, individuals who inherit one copy will express the disease phenotype. An example of an autosomal dominant disorder is neurofibromatosis type 1. This disease causes tumors to form in the nerves and skin. If a parent has an autosomal dominant disorder, there is a 50% chance that each child will inherit the disorder. An X-linked recessive disorder occurs when an abnormal recessive allele is located on the X chromosome. These disorders are much more common in males. This occurs because males only have one X chromosome. If a male inherits the abnormal allele, he will express the disease. Females would need to inherit 2 abnormal alleles to develop the disease. Females with only one abnormal allele are typically carriers and do not show symptoms. An example of an X-linked recessive disorder is hemophilia A. Hemophilia A is the most common form of hemophilia, a disorder that impairs the body's ability to form blood clots. If a carrier female has children with an unaffected male, 50% of the male children may develop hemophilia, and 50% of the female children may become carriers. If a father has hemophilia, all of his female children will inherit the abnormal allele. However, his male children will not inherit the disease from him, because sons inherit the Y chromosome from their father. For a female to develop hemophilia A, her father would need to be affected, and her mother would also need to be a carrier. Another inheritance pattern is X-linked dominant inheritance. An example of this disorder is vitamin D-resistant rickets. This disease causes soft and weak bones. If an affected individual has children with an unaffected partner, there is a 50% chance that each child will inherit the disorder. If the father has this disorder, all of his female children will inherit the disease. However, none of his male children will inherit the disease, because sons inherit the Y chromosome from their father. Understanding these inheritance patterns helps clinicians estimate genetic risk and helps families understand how genetic diseases can be passed from one generation to the next. Dominant, recessive, and codominant allele interactions help determine an individual's phenotype. Punnett squares can be used to estimate the probability that offspring may inherit specific traits. These genetic principles allow us to predict how autosomal dominant, autosomal recessive, and X-linked disorders will be passed from one generation to the next.